In this last video for the microbial growth and nutrition lecture, I'm going to discuss microbial growth and enumeration. So bacterial are asexual reproducers, and essentially what they do is similar to mitosis in human cells, which essentially is just making a copy of the genetic information and then splitting the cells in two. So you create two cells that are genetically identical, and this is a process called binary fission. So when nutrients are fully available and we're able to make enough cell wall and cell membrane, that will then initiate a copying of the chromosome, you see here and here, which we'll talk more in detail about when we do microbial genetics, and therefore the cells start to cleave and separate into two, and so then we have two cells that are exactly the same. Now, as a result of binary fission, the population growth is actually an exponential growth, because after one life cycle or generation time, it goes from 1 to 2 cells, and then 2 to 4, and 4 to 8, and 8 to 16, and so on and so forth, dependent on the cycle. The generation time for organisms is typically very rapid when conditions are favorable. Anywhere from 30 to 60 minutes is an average time, and E. coli is actually only a 20-minute generation time. And doubling will just continue as long as all of the nutrients are available and the conditions are right. So it's important to be able to establish a population growth curve for any organism, knowing exactly sort of how they're growing during a time frame where they have access to nutrients. And this becomes important when applying them to drug regimens, but also when doing research with the organisms. So this is typically done by placing a small amount of bacteria in a certain location, aseptically into maybe a tube or a broth of some sort, and allowing it to grow under favorable conditions, and at certain time intervals during the incubation period, then counting the number of bacteria that are present. Now, there's lots of different ways in which we can go about and count bacteria. One of the most common methods is spectrophotometry, and this is basically measuring the turbidity of a broth. So if we were to look, and turbidity just means cloudiness, right? So this right here is a spectrophotometer, and this is actually an indirect cell count because you're not counting every single cell, but you're just looking at the cloudiness and using that to infer the number of bacteria that are there. So a nice fresh broth is this color, and it's very clear and easy to see through. And if you put it into the spectrophotometer, you will shine light, and all the light will be able to be transmitted through that very transparent liquid there. So it measures transmittance, right? Then as we allow the bacteria to grow, the sample gets cloudier and cloudier and cloudier, and light ends up being reflected off of the bacteria, and therefore less light is transmitted. And so we can equate the cloudiness to the number of bacteria that are there, and that's an indirect cell count. Now, we can also do a direct cell count, and there's different methods for doing that. One method is hematocytometry. So hemato means blood cell counter, and so typically this is used to do white blood cell counts or red blood cell counts and things like that. But we can also use it for bacteria, and it requires a special slide, microscope slide, called a cytometer that has a very small grid on it, and you put an exact sample size on there. And then you can count the bacteria within the grid and use statistics to make an assumption about the number of bacteria that are in the bacteria that are in the overall sample. Another way we can do it is by either a Coulter counter or a flow cytometer, and these work in similar ways. We actually are able to design pipettes that are very, very tiny, and flow cytometers are actually about cell size, so they only allow one cell to pass through at a time, and so we are able to count as those cells pass through using an electronic device. Flow cytometers are specific to being able to differentiate between live and dead cells, and this is important being that we might want to distinguish or be able to count how many bacteria are alive or dead, say if we're trying to test a drug or something like that. These are actually very expensive pieces of equipment and aren't very realistic for using in the average lab. Because they are so expensive, they might not be available everywhere that you are. So as we count the bacteria, then we plot those bacteria onto a curve. So you see on the bottom here is time, and on the y-axis here is numbers of viable cells. And this is a logarithmic scale, so it's been changed so it looks linear because this is a log scale rather than being a curve, an exponential curve. So there are 4 different phases of a bacterial growth curve, and the first is a lag phase, and this is when the cells are just adjusting to the growth. So the numbers stay pretty steady for the number of bacteria that you have in the sample at that time. And again, remember, we're taking out a sample at each of these time intervals and actually counting how many are there. Then we have the exponential growth phase. So the exponential phase is when you're at the maximum growth rate, they have full access to available nutrients, and we're seeing that exponential growth of 1 to 2, 2 to 4, 4 to 8, and so on and so forth. After a certain period of time, then nutrients will start to deplete and space will start to run out, and we enter into a stationary phase where death is actually going to equal the rate of division. So some organisms are dying, they're not getting nutrients, or and then they're polluting the environment and killing off other bacteria, whereas some still have access to nutrients and therefore are dividing. So we sort of flatten out. And then ultimately, over time, the environment starts to become toxic, and so there is this 4th phase where we actually see the bacteria dying off as a result of toxicity and loss of nutrients. Now, growth curves are important because they can be used for determining the different stage of growth, that can be used in research, trying to look at drug regimens, how frequently you have to take drugs. Every 4 to 6 hours you take this dosage, and also understanding the different stages of growth when looking at how actively they are metabolizing things in their environment, which could be important for studies.